Hi there, I'm Professor Rob Bristow from the Translational Oncogenomics Laboratory at Cancer Research UK's Manchester Institute. My laboratory currently consists of 4 graduate students. They're doing their PhDs looking at the role of either oncogenes or tumor suppressant genes or DNA repair in the context of hypoxia and genetic instability. We have a senior research officer that is driving the overview of the lab, but also specific conditional expression systems with respect to DNA repair mutants, and then also a postdoctoral fellow who is driving our bioinformatics program and including looking at a whole genome sequencing of prostate cancers and trying to understand how genetic instability is reflected within those specific bioinformatic changes. My laboratory also has a number of clinical trainees. This includes clinical fellows, medical students, or residents who are working on their own projects pertaining to the translational aspects of prostate cancer biology. And this includes using primary specimens along with our pathology colleagues to look at in situ DNA repair and chromosomal instability. The actual vacancy itself pertains to the role of these DNA repair genes in terms of driving genetic instability in relation to other oncogenes or tumor suppressor genes that are either activated or inactivated during the course of progression. So you'll be utilizing and driving novel primary prostate cancer epithelial models that are turn immortalized and then either knocked out or knocked in for specific mutations associated with DNA repair, particularly genes that are associated with double-strand break repair, such as BRCA2, ATM, or non-homologous end joining associated genes, and then also mismatch repair, such as MSH2. In understanding, again, the genetic instability, you will be driving forward a number of DNA repair assays and chromosomal instability assays in vitro, and for those cells that become transformed when we knock out further genes, P53, MYC, PTEN, then looking at transformation assays and actually growth of these cells in vivo. There's also an opportunity to drive forward a model looking at, again, the interaction between DNA repair and oncogenes within a genetically engineered mouse model. The CRUK is a fantastic ecosystem for science. A number of the basic and discovery labs are driving forward a primary cell and molecular biology as well as functional genomics. And therefore there is exquisite and state-of-the-art core facilities with respect to spatial transcriptomics, proteomics, genomics, and then also screening facilities. There's also, of course, a number of collaborations with our clinical colleagues in which the CRUK core facilities actually are driving forward a number of the translational studies in which we look at a number of these changes that I've described within primary prostate tumors in situ. Manchester itself is a fantastic city. It's always growing. It's always a buzz to it. It's a fantastic music scene, great clubs and restaurants. And if you're an outdoors person, or surrounding Manchester are fantastic areas to hike, to bike, and also to visit for the day. This includes the Lake District, the Peak District, and some of our great national trusts that are around Manchester. And of course, Wales itself, which is only an hour away, and therefore only the ocean is an hour away. So if you're interested in this position, please feel free to contact me, my laboratory. And of course, after that, put in an application. We look forward to seeing your CV and the reason you'd want to join our lab, and happy to talk about further details in due course.